Thanks for the introduction. Let me share my screen and start my presentation. So, first of all, really thanks for the organizers for creating this meeting. You can see the slides, I assume, right? I'm trying to see. Yeah. All right. Perfect. So. Yeah, and well done for getting so many people to come to this. I think leaving computational out of the title must have worked. Or maybe this is just a topic that many people find very relevant. And it's true that it's a topic where there's so much happening that it's difficult to figure out what you need to do. And a little bit of help, I think, is appreciated to get you started. And I'm also looking very much forward to the next two talks and maybe learn a bit more about this topic myself. Now, I want to talk a little bit about the importance, the basic importance of computational reproducibility. So the main goal of computational reproducibility is, it sounds relatively unimpressive, but it's surprisingly difficult. And that's if you use the same data that you used to get some results, after a while, or if you use the data and the code of someone else, you're supposed to get the same results of analysis. That is basically what it boils down to. So you're not even creating anything new. You're just creating the same thing that somebody else did, with the goal, of course, to either be able to check what they have done in their analysis, or maybe also to tweak it. One reason I appreciate it if I get to review a paper that is completely computationally reproducible is that as a reviewer, I often have some questions like, what would have happened if you've done this or this? And if I get a submission where I can just go into the code and run the analysis again and check, I don't have to ask. So I don't have to ask you to revise and resubmit something. I can just say, oh, look, I already checked. It's fine. It's fine. Don't worry about it. So I like it. The language is sometimes a bit confusing, and I have to apologize being part of, at least in psychology, a little bit the reason for this confusion. So in 2015, we published a paper where we replicated 100 studies in psychology, and confusingly, we called this the reproducibility project, which really didn't make any sense. But what did we know in 2015? I mean, we were completely clueless about this. So we should have called this, of course, the replicability project, because here we were trying to replicate studies. So we collected new data and we tried to find the same results. That's not what we're talking about. So we're really sticking with the original data and trying to find the same thing. Now, it's a modest idea that if you analyze your results, that you make sure that your workflow, your scientific workflow, has this step of computational reproducibility in there. And I think it should be a minimal requirement for any scientific workflow that you have. And again, even though it is a minimal workflow, it doesn't mean that it's easy to do. Now, I don't know how your experience with computational reproducibility is, but I can share an intriguing finding. And I think scientifically, it has not been completely explained. The underlying mechanism of what I want to talk about has not been explained. I'm not sure that scientists know how this works, but it's an effect, and you can notice this effect. It's called data rot, and it happens in a very specific situation. Namely, it is a process that occurs between submitting your work for review and then doing the revision. So it works like this. You analyze your data and you find some results that you're very happy with and you think are important enough to share with other people. And you do your complete analysis and you write your paper, and then, you close all your files and you submit your paper and you wait a couple of months, and then people have reviewed it and hopefully they like it. But very often they have a couple of other questions. They would like you to maybe add a bit of an analysis or plot something that you didn't plot before. And this moment for the revision, you open your data set again, and there it is, this weird process of data rot. And it means that no matter what you really tried before, it's impossible to get exactly the same results as you actually originally submitted. And now sometimes through sort of reconstructive processes, you can get back exactly the same analysis that you submitted. And it's very often a long and difficult process. Data rot is a really sort of nasty process, nasty thing to try to prevent. And often it works out in the end, but you go through an extremely stressful process. And I'm really serious. Like, I mean, I know that this part is a joke, but the stressfulness of the process is not a joke. So I really very often had this situation that you open your data set and the first 30 minutes, you're just like, oh my God, oh dear, I don't know. What did I do? Is everything wrong? Is everything right? And the moment that you realize everything is actually hopefully fine, this takes a while. So computational reproducibility, in a way, you know, we're asking people to do a little bit more work than maybe you're used to, but there are some direct personal rewards, and this lack of stress is a big plus, I think, a very important part. So although the aims of computational reproducibility are modest, we don't really do a very good job at this. And I think the reason for this is that it's just new for most of us. Now, I know that I am by now particularly old, you know, I understand. I mean, I'm 40 now. So what does it mean? It means that, you know, I created an Hotmail email account when I think just before it was bought by Microsoft. And I downloaded music on Napster, and I downloaded punk songs that you could download in about 30 minutes because you could download one song. You know, this is the internet that I grew up with. This was the limitations that you had. And you have to keep being dialed in, of course, because if you break the connection, you can't resume a download. That's impossible. So this is what I was brought up with. And well, many people that you work with might even be older. So, you know, their experiences are from when they were young are even different, right? So their experiences with computers. So the development, like, I'm still amazed that, like, what we are doing now, for me, still, you know, I'm still amazed at the ability to be able to interact with so many people over a video stream. The fact that you can store data for free somewhere is amazing for me still, that you can just upload your data somewhere and somebody will take care of it for, I don't know, maybe like at least a 50-year guarantee. This is all amazing. So it's relatively new, and especially for scientists, this is new because science is really slow. You know, science develops over time, but not in very rapid way. So for many people, this is new. So we all have to learn how to do this. And one of the issues is that we have not developed a lot of standards, for example. You know, we still have to figure out what good approaches are. And I'm really happy that some of the talks here are going to talk about this in more detail and introduce you to some standard workflows for this. So it's new for all of us, so don't be worried. I mean, the Twitter and the internet is a good place to ask. There are many people there that you can ask for questions about this. But don't be worried that you don't know how to do this, or that you're going to, you know, maybe make a mistake here and there, because that's also all part of the process. If we take a look at the current state of the art, and I really think this is, some of these things are state of the art in different ways. So I have a couple of papers where people tried to computationally reproduce papers that were published. And all of these papers, you'd have some good hopes that it would have been easy to reproduce these for different reasons. In this paper by Tom Hartwig and colleagues, 2018, people were required to share their data. This was a new sort of a requirement of a journal. They started to ask people to share data. And well, they thought, okay, let's take a couple of these data sets and just try to create our own code and reproduce the results. So this is just from scratch, create new analysis. So this is not computational reproducibility yet, but it's just, can we get your same results? So they first tried this, which is very difficult. And then after this, they contacted all the authors and they asked for help. And of the 35 articles they had 13 sets of results where even if they asked the original authors for help, they couldn't reproduce the data that were reported in the paper. So this is, you'll see that these numbers keep popping up. So these percentages are not surprising. This is sort of where we are at. In another paper, people looked at publications from political science from 3 different journals in 2015. Here they first had to ask for the data, because, well, I don't know how many of you always share the data alongside your publication. Would be lovely if all of you do this, but it's not yet common practice that data is always shared with the publication. It's not required. So here they asked for the data. They got it from 71 articles after emailing the authors. That's nice. I mean, these were relatively recent articles, so people didn't die yet, which is always easy to, you know, if you want to get a reply from somebody, it's always useful. We know that these percentages drop over time, of course. So for this specific case, for one article, they couldn't reproduce the analysis because they didn't have a software license. We had a similar experience. I know, I mean, it's peculiar maybe, but I don't have all software that exists. We don't have this as a department. So I'm sure that other people later on in this meeting will also talk about the importance of using open source software where possible. So here they had 16 articles where they could not reproduce the results out of these 71. 32 could be exactly reproduced. So that's about, 50%, a little bit less, but it's pretty good. 19 could be reproduced with tiny differences, and 3 articles really yielded different results than were reported in the paper. So this pattern, again, you see it more and more often. This is a study where they looked at articles published in Science, and maybe we want to assume that this has pretty good probability of being computationally reproducible because, I don't know, high profile outlet. So people might have taken very great care. Here, about 26% of the data sets that the authors acquired could then be computationally reproduced. So that's not that good as well. We ourselves this year looked at a subset of articles. We were interested in all of this is new for many people, but what are the people doing who are sort of embracing state-of-the-art stuff anyway, early adopters. So we looked at a specific population of findings, which are registered reports. So in psychology, we have this new publication format, registered reports, where you can submit your research before you collected the data. So basically your theoretical introduction and your statistical analysis plan. And the reviewers take a look at this, and if they like it, you can go and collect the data. But this is a process where we thought, okay, these people like all the new stuff, they like to do the sort of open sciencey things. So let's see how well these people are doing in terms of computational reproducibility. Now, an important difference here is that we didn't email people for their data sets. We just checked how many data sets are available. So this is really without any help from the authors. How far could we get? And the numbers look, given this property, a little bit better. So there is definitely something about these people who are embracing registered reports and early adopters. They're trying and they're doing a little bit better than the numbers we've seen before. Even here, out of the 62 registered reports we were able to look at, 41 of them had data available. That's pretty good, right? So these are people who out of their own free will already share their data. 37 had analysis scripts. And we could run the scripts for 31, so that's half, and reproduce the main results for 21 articles. So even that, this is the people who are really trying, I think. And here you could see some really good examples. You could also see some reasons why it's difficult to reproduce articles. And finally, there's a more recent study, even more recent. And these are papers where you'd also hope that they were reproducible because these people received a badge. So there are now these new badges that you can get, sort of like kindergarten stickers. It's not really about the sticker, actually. I think there's this idea behind these badges that hopefully in a while, if you don't get a badge, people will be, like, why didn't you get a badge? What's wrong with why didn't you do this? So that's the goal, I think. But okay, these got badges, so the data was openly available. And here, again, 36% could be reproduced without the help of the authors. And then 28% could not even be reproduced if the authors were involved. Now, this didn't lead to large conclusions, right? That's important about these data. It's not that just because you can't reproduce a number, it means that the data is invalid or the paper is invalid. But we still would like to have a situation where I can find the same numbers and the same data that you wrote down in your article. Maybe I want to reuse it for something else. Maybe I just want to check if everything is fine. And I already mentioned that sometimes it's useful to take data and do new things with it, right? Okay, so this is the current state of the art. So don't feel bad if you're not doing a good job. You're just, everybody's trying. There's clearly room for improvement all around. So, and again, I don't think this is weird. So I always, sometimes I think about things a little longer when I'm walking my dog, for example. I think about these things and I think, it would be peculiar if this would be the time where as scientists, we have figured out how to do everything perfectly. That seems to me just extremely unlikely that we happen to live in a time when we figured it all out. So there's always stuff to improve. I'm talking here about stuff. And I think my hope is always that 10 years from now, people like you are going to say, like people in the audience, you're going to say, oh yeah, I heard this talk and this person recommended this, but that was so crazy 10 years ago. They were so silly about it. Of course, we don't do it like this anymore. We do it like this now, right? So then you can educate me about new things. And that's how this all is supposed to work, I think. Science, I mean. So there's a lot of room to improve. And I think some of the things where we really want to see some improvement is in the computational reproducibility, but also in terms of the metadata that we provide. So for starters, this is just your code book. And I know the code book is the most boring part, you know, I mean, really, really the most boring part you can do about research, where you make your code book and you explain exactly what's in your data. But it's super important to provide this kind of data with your article. It's also useful to develop structures in which we store things, like what is a good folder structure, so that if I look at something, you shared your data and I look at it, I actually know where to find stuff. A large amount of time when we try to reproduce these registered reports, just clicking around in folders and like which file is going to do which thing, and where's the pre-processing of the data, and where's, you know, everybody has their own sort of folder structure. Many people don't have a folder structure. They just throw everything together, you know, but this is also something we can improve. Some fields have a pretty nice setup and are a good inspiration for people who want to develop these kind of rules, like bits. So this is for brain data. They have a very nice structured format and also a folder structure. So we can learn from this. There's an initiative that I really like that's trying to do this for psychology data overall. So PsychDS, still ongoing, not done. You see, people are still working on these things. And there are more of these examples, but metadata is also important. I think there's a bit of a depressing thing of knowing that even now we're going to do our best to share our data in a very reproducible way. And it is lovely in the sense that if I am very interested in your specific data, if you do everything well, I can go into this data and I can reanalyze everything and I can figure out what you've done. But what we actually want to do is also be able to find stuff across data sets. So search metadata and then go into your data, right? That is the bright future that we're working for. So this is a first step towards this, making sure every individual project is well organized and reproducible. And then if we can also look across data sets, that would be super efficient, right? So we want to, in the future, hopefully combine metadata and computational reproducibility. And I think we can do some cool stuff with this, right? Because then eventually I would be able to find your data and your hypothesis and reproduce them, for example. So we have this recent paper where we play around with this idea of a sort of ideal organized scientific data structure. So we call this science verse, which is of course a bit of a, you know, reference to well-organized sets of things like the tidyverse, for example. And but there is a very old idea from the 30s where there's some sort of grammar of science. So we should be able to organize everything that we do as scientists in well-organized blocks and give it all a name and be able to find it. And we talk about how you can organize your data. If you have a well-organized data file and a computationally reproducible analysis workflow, then I can basically go into your entire folder, just grab all the information in there and run your analysis for you to check what you found, which you can, for example, do if you want to check whether a pre-registration. So you pre-register your predictions. You say, this is what I predict. If you have a well-organized data file and a computationally reproducible analysis, in the end, what we want is that I can just click a button and run this analysis on your data file after you collected the data to check if your predictions worked out in a pre-registration, for example. Or maybe if there's good data names, I can even run the analysis for something that I'm meta-analyzing and grab the effect size. Now, this is far, far in the future, right? But I think it's useful to imagine a future where we'll have data like this and the stuff we'll be able to do if everything is well organized. Now, I'm going to be dead before this happens, but you might not be. And I don't know, depending on how well these excellent other speakers of today are doing their great work, maybe I'll still be alive. Maybe I'll see some of this, but that's, I think, the end goal. And then we can just have real access to this huge, rich data that we all collect. Right, we can actually reuse it. So this is a bit of the negative thing, that we have to agree that we're trying to organize everything well, and it's very likely that 10 years from now, when we've really figured this metadata out, we have to sort of start again. That's okay. I mean, then we have a really organized file system, but now we can still go in and run your stuff. So that's a good start. Now, if you want to get started, there's a lot of things to consider. There's a lot of like, what are you going to do? Which tools are you going to use? And there's a huge amount of stuff that other people will also mention in their presentation. There's a huge amount of stuff to consider. And the problem with everything being so new, and also software packages coming and going, is that there's no single standard. So you can't ask like, okay, what is the way we do this? I have a workflow that sort of a lot of my collaborators drift towards, which, has version control on Git, and we use R for analysis, and we write our papers in R Markdown. Now, this is our workflow, but there are many other variations, of course. So we have to drift towards, some sort of preferred solution, which probably differs between sub-areas. So take a look around and, look what other people are doing. There is an important thing. So now you're like, you're still enthusiastic. You're like, oh, this looks good. I can do this. We can make science better. And let's contribute to making science better, right? You might be a bit motivated to do this. That would be lovely. I do want to point out one thing that I think we also need to figure out. And it has nothing to do with computers, but it has a lot to do with people. The moment that we start to share things very transparently and we make all our analysis reproducible, it means that we can also perfectly reproduce all the mistakes we make and all the silly stuff that we do. And that's pretty scary. I mean, I have tenure and I find this pretty scary, but many of you might not have tenure and I find this even scarier to do. So we'll have to learn to accept. And the important thing is now it looks like nobody is making mistakes. I mean, of course, that's ridiculous, right? If you ever started your computer and it says, I am installing updates, that is just fixing mistakes that other people made. But you never open a scientific article and then it says, oh yeah, we're installing some updates to this scientific article. We're fixing some. No, never happens. We always pretend that everything is perfect. That's crazy, right? We all make mistakes, but we just don't share them. So we'll also, as we move towards this more open and reproducible workflow, we'll have to learn to deal with the fact that other people are going to find our mistakes because we're sharing those as well. And that is going to be scary. So we have to learn how to have this as a culture transition as well. And I think the older we are, the more we should say that we make mistakes all the time. I think that's the easiest. For me, it should be much more easiest to say, and then hopefully younger people feel more comfortable with this. But this is going to happen, right? This is a sure thing going to happen. There are also some things we have to organize on the level of universities, or maybe the way that your lab works or your department works, where you don't have immediate control over, but it also needs to happen. One of my favorite groups that is doing these kind of things that you might want to look out for at your university, wherever you work, and if it doesn't have one, you might want to start it, is an open science committee. These are people who are helping the whole university figure stuff out with data privacy and what are good ways to share data, but also, very importantly, what should our data informed consent form look like so that we can share data? Because you can have a reproducible workflow, but if you'd never asked your participants if you can actually share their data, then what is this going to run on, right? So you can't. And it's not that it's impossible to do. It is very well possible. There is a link here at the bottom, or at least this research data management support banner is from Utrecht University. So I'm from the Netherlands. They have a great website guiding you how to do this. There are more sources like this, but it probably means you have to do something on your department as well to make sure that you can share this data with others. So this is just this default sentence that they recommend you add to your informed consent form. Take a look around what other people are using, but that's also important to do. So I already mentioned in the beginning that I really like to get reproducible files. So you get some benefits. That's my main thing that I want to say. You get some benefits from this. Like I really like to see data. It takes away some of my doubts that I sometimes have. It helps me answer some questions that I'm not going to bother you with if I can just check it for myself. Assumption checks, for example, I'm crazy enough to sometimes want to look at those, and then I can just see everything's fine. So it can help you also in terms of the quality of the peer review, right? It can help you better if I can take a look at everything. Make sure that if you share it, other people can reuse it so that you add a license to your data. That helps scientists know what they can do with the stuff that you share. People often forget it, but it's pretty important. And make sure that you upload the stuff that you share somewhere where it's stable. So your personal website is not going to be sufficient because, you're not going to work at the same place you work now 10 years from now. Maybe you will, but likely you won't. So nothing is forever, but there are some repositories, and you can ask your information specialist at your library typically what this is. So we still tend to think of librarians as if they do stuff with books, but you know, they also developed over the last 20 years, and they are hugely important resources for a lot of this stuff. And very often they function as a sort of guerrilla warriors within a university to, you know, improve open science. They're, in my experience, all of them are great people. So reach out to your librarians, help with these kind of things. So I already mentioned the boring parts, like making sure you have a code book, make sure your data is fair, all these kind of tiny steps, but all of this together is needed to really make it possible for other people to reproduce your analysis. So this is an important skill. Prepare that you have to train it, and you're going to learn a ton of things only here, right? So it might be a little bit overwhelming, but I would just recommend start somewhere. You know, pick something that you hear here today that you like. Just start with that until you feel comfortable, and just take one step at a time. And then eventually you'll get there. All right. Thanks for the attention.
Great.
Thank you so much for that introduction. I'm Adina, and I indeed work at the intersection of psychology, neuroscience, and computer science in an exciting field that is called psychoinformatics. And whenever I can, I actually also work computationally reproducible. So in my talk, based on experience and concrete examples, I want to highlight some of the specific issues in neuroscience that make computational reproducibility challenging in my field, but I also want to highlight some of the solutions that exist in the field and that make me able to work computationally reproducible. So I want to show you a recent paper of mine. Here's how that looks like. What you can see here is a publicly accessible GitHub page. What's important about this is that anyone, anywhere can access and retrieve this paper. And I've said that this thing is a scientific paper, but this collection of files in here, it does not contain a PDF. Instead, it proclaims to be the code, data, and manuscript for this publication here in this recording, which is an actual PDF with plenty of numerical results and figures. Now, when you or I or someone else gets themselves a copy of this repository, they can follow a few steps in the instructions that this README provides. And those steps list the necessary commands to get the relevant software installed and instruct the reader to run a single command, which is currently happening in a very sped up fashion in this recording. And the single command is make, and Andreas will later talk a lot about this, I think. So what happens here is that with the execution of that make command, a cascade of things is triggered. All data, however large this data may be, is downloaded from wherever this data is stored in the precise version that I specified. All of my analysis code is run on this data to produce the results and figures. And those results and figures are dynamically embedded into the manuscript just as they have been computed at this very moment. And afterwards, one has the dynamically generated manuscript as a PDF on their computer. And here you can see me actually opening up this paper. It has all of the figures, all of the data. And if I extend the code now with additional analysis or fix a bug in my code, it takes only a single command for me to update my paper with new results. I also want to show you how I do data analysis. When I do a computation, I use an open source language instead of a graphical user interface, and I write scripts that hold my analysis code. I think many people do this, and this is a very good thing to do. And the execution of this script looks a little bit unfamiliar at first sight, but what I do here is I execute the code in a way that a machine can store a record of the complete execution. And that record includes a precise specification of input data in precise versions, a precise specification of the version of the code that I'm using and how I'm executing it, a complete specification of a software environment to use, and a complete record of the resulting files and their precise content identity. And what I get from doing this is a digital record of what exactly has been done, when, why, by whom, on what data, and in which software environment. And that record is attached to whatever result my script execution has produced. And it is identifiable with a unique identifier. And when I want to redo a computation or verify that a previous computation produces the exact same outputs, or if I want to share my project with collaborators, then I can find that unique identifier of the electronic and machine readable record and plug that into a command that tells my computer to automatically redo all of my previous computations and automatically verify whether the content of the resulting outputs is identical between the two executions on my computer or on another computer. And if the content is not identical, then I have the tools to investigate where precisely the differences in output lie. And that's quite nice. Now, why do I work in this way? First and foremost, I do it because it makes my life a hell lot easier. It allows me to enable my future very forgetful self and also others to understand, redo, and verify what I've done and build up on it. It makes my research better, definitely. People can find the mistakes I make. It makes it more accessible and adaptable. And it makes collaborating with others and my past self much easier. But in order to work computationally reproducible in neuroscience, there are a couple of obstacles to overcome, and I want to highlight a few of these issues to give you an idea of them. The first specific issue is related to data. Whatever type of neuroscientist you are, you'll acquire or work with interesting data that can have some of the following peculiarities. It can come in proprietary formats that require expensive software. It can be very large. It can be very heterogeneous. It can come in sizes that exceed the storage capacities of your computer. And finally, the data that you may work with could fall under GDPR. And all of these data issues can make it much harder to access data, to structure it in an understandable way, or share it publicly. But data is unfortunately not the only issue, because it's also analysis. Regardless of what you are researching, all of our lives would be so much easier if we could go from data via a bit of analysis directly to our open access paper. But in reality, it usually takes a sequence of analysis steps from the raw data to the final result. And within each of those steps, there is a large stack of software and analysis methods to choose from. So typical analyses are complex, multi-stepped, and they contain many researchers' degrees of freedom. And to just illustrate this complexity and the analytic flexibility, consider an influential paper from my field that came out earlier this year, the NARP study. In this study, 70 independent research labs were given the same raw data and the same 9 hypotheses to investigate. And the result of the study was that those 70 research labs each came up with their own complex analysis workflow. And based on these 70 different workflows, they actually only drew consistent conclusions for 4 out of the 9 hypotheses. Now, with regard to computational reproducibility, it's not the variety of methodological and analytical choices that's the problem here. The challenge for computational reproducibility is to encode all of those researchers' degrees of freedom that led to those 70 different analyses and conclusions in a standardized and ideally machine-readable way. And the reason for this is that the building blocks of a scientific result are rarely ever static. Whatever makes up your analysis workflow, it is certain that it evolves. And the most straightforward example for this is the code that you use to analyze your data. In the wake of your study, you will likely find bugs. You will add code or function after a review. You will refactor your code to make it more readable and so forth. And you will want to know which version of your script was used, because changed code usually also means changed results. But it's not only your code that changes, it's your data too. In almost all analysis that I have done, the data I used evolved. I found errors and I fixed them. I reorganized the way that the data was structured, or the standards changed to which I had to reorder my data to. I created derivatives, I renamed it, and so forth. And to really bring this point home, here's a concrete example. In the computationally reproducible paper I demonstrated at the start of my talk, I had to deal with changing data. And the reason was that in the end, when all analyses were done and I wrote the discussion, I found an error in one of the input data sets that I used. And the error was reported in the literature. It was an open data set, but it was not fixed in the available data. So what I did was I submitted a fix to that error. And you can see a digital log of that in the screenshot here. And what this shows is that a researcher uploaded their data in 2018, and in 2019, I came around and fixed that data. Now, what this means is that the data set from 2018 and the one from 2019 differ. And changed data means changed results. And if I don't have a way to record the precise subset of data and precise versions that was used in the data analysis, then the computational reproducibility of my work is threatened. Now, with the set of tools that I used in my publication, it was actually very, very easy to deal with it because I could update the input data, I could register its version. I had written the paper dynamically generated, which will be something that Andreas will demonstrate later, I think. So it only took one command for me to recompute everything, update my results and figures, make a PDF out of it and submit it. And that's certainly where my life got so much easier. But typically, it's this complexity of data analysis together with the evolution of the building blocks of a study that create these acute cases of, shit, which version of which script produced these outputs from which version of what data? And that usually happens just before the deadline. Because there's typically a really complex and multi-step manual process that generates results. And throughout the lifetime of a project, that process is actually repeated many times. And no one outside of the project, and even the people that are involved in the project, can remember how exactly a result came to be, or which is the most recent one, or which is the correct one. And that's why we need to make this analysis pipeline transparent so that it is clear for us and others which version and subset of which data has been used with what analysis and what parameterization to create a result or figure or the PDF. And the term for this kind of information, a complete description of how a digital file came to be, is digital provenance. And that is a fundamental piece in fair, so findable, accessible, interoperable, and reusable science. It's part of the reusable part. But beyond transparency, the pipeline also needs to become automated, because anyone who does not know your project and even your own future self will not know or forget all of the relevant steps in the labor-intensive manual process that creates a file or result. And that's, for example, why I not only use scripts instead of graphical user interfaces, but I also bundle up all of the steps together in a single workflow file, such as a makefile, which Andreas will talk about, just as I have done with the paper that I demonstrated. And yet further, unfortunately, it's still not done because we have also issues with our tools. Software is a really important part of the digital provenance of a result. You know that if you've ever tried to recreate a result without having the software installed or having a wrong version installed. But in some fields, the required digital provenance goes beyond one software because modern data analysis software has an incredibly large and complex dependency stack. Here's an example, and you don't need to see the details in this figure. Just note that every box in this graph is another package or software library that is a required direct dependency to make the package in the top corner, scikit-learn, work on your computer. That's quite substantial. But here is the dependency stack of R base core. And here is the absolute dependency hell of JASP. And this could make the most boring fun fact that you have learned today. But unfortunately, this is stuff that matters. Here's a paper from Tristan Glattard, who tried to rerun the same analysis on the same data on different operating systems. And he failed to computationally reproduce the results. And he really dug into the reasons for this. And he found multiple reasons. But one case, for example, is that version or implementation differences in very, very basic underlying software libraries, those that enable your computer to do the math, those produce numerical differences. And those differences may be small, but in many fields of neuroscience, we do things a couple million times, and almost no one has a one-step pipeline. And in those cases, those numerical imprecisions add up. And this is one of the reasons for the works on my machine problem, where an analysis works on your computer, but it produces a different result or fails to run it all on your friend's computer. But this is also a reason for this used to work on my computer problem, where an analysis from 6 months ago either does not run anymore at all or leads to different results. And in both of those cases, where an analysis does run but produces different results, how do you decide which numerical result is the correct one now? There is no way. Now, all of these issues and the measures that we should take as a consequence of them typically exist on top of the complexities that we all have to face anyway in our scientific careers. We are all probably trying to learn how to code. We have to teach, acquire data, graduate, get tenure. We have an impatient advisor or multiple projects to juggle. And also, all of us in many cases have no formal training in computing or research data management. So that's really a tough thing to handle on top of that. So in many cases, the final result that gets published is just a teeny tiny block in an incredibly fragile stack of things that are not only complex to do anyway, but also have an impact on the computational reproducibility of the result that we are often even not aware of. Be that software, software dependencies, data that changes, analytic flexibility that isn't accurately captured, or just social or cultural pressure or stress. So, which solutions exist in neuroscience that get us towards computational reproducibility, despite all these issues? I want to highlight a few aspects that I find relevant. The first one will be open source software. The next one are non-neuroscience specific solutions to problems that also the industry has. And the last one is the impact of training and supportive environments. But what I want to really stress first is that in a heterogeneous field like neuroscience, there is no single one size fits all solution. Instead, there are many different individual solutions to all of those individual problems, and we can connect them however our individual field requires to make it work. So the first solution that I want to point out is open source software. One thing that is great in neuroimaging as a subfield, but also in other subfields of neuroscience, is that there is an open source solution for basically everything. We have open data standards and open source tools to do science, and we have a community that heavily contributes to the development and improvement of those tools. And using an open source solution may mean more work, as you may need to learn to use the tool, but open source removes the accessibility barriers to computational reproducibility, and it allows to make software a transparent piece of digital provenance in an analysis because it is open source and because everyone can install it. But now to a few tools and concepts that the industry is using to solve similar problems like we do in reproducibility management. The first of those concepts is version control. And put simple, version control can make a complex project non-chaotic. Version control allows tracking changes that are done to files over time and identifying precise versions of those files uniquely. So instead of having a project directory where you have 20 almost identical files and only slightly changed file names, version control lets you have an organized directory with a history through which you can browse, identify specific versions of files, or that you can use to reset files to specific versions. And while they do this, version control tools collect, store, and share digital provenance of what has been managed by them. So the screenshot that I showed earlier that demonstrated changing data, that was a version history taken with Git of a dataset, and I was able to uniquely identify a dataset version with the help of this tool. A second set of special tools or concepts that I want to highlight are related to the installation, dependency management, or packaging of software environments. For one, there are software repositories such as NeuroDebian or Debian Med that package up complex, complete software and their dependencies, and they provide convenient installation and dependency management. Even if you have never heard of them, they are very useful, and if you've ever used a software container, then they come of use there. And here's the second thing, I've mentioned it already, software containers, which are portable software environments that allow you to run whatever software is inside, isolated from your own operating system. And this is very useful if you want to encapsulate a complete software environment with very specific software. For example, if you have a very complex software stack in your analysis. So these are the tools that can open up the way towards capturing also the software provenance and defining environments for reproducibility in the way that we work. But the next step is also really hard, maybe the hardest, because the next step towards computational reproducibility involves how to handle data with whatever issues it has. First, the potentially large or complex datasets or analysis that we all have, regardless of the subfield that we're in, they all require very good research data management from the very start, from the very first time that you work on a project. data needs to be handled according to those FAIR principles that, Daniel mentioned, not only to maximize the impact and potential of that data, but also to make computational reproducibility possible in the first place. And both research data management and reproducibility management are not only tightly intertwined, they also have in common that they really can't be an afterthought. You can't go to a project in the final phase and apply all RDM principles retrospectively, and it's also almost impossible to make a project computationally reproducible only as the very last thing. And because RDM is that hard, especially in neuroscience, there are national and international initiatives that improve RDM and provide tools and infrastructure and education for data sharing and reproducibility management. And just as Daniel has already said, there's a large range of infrastructure for sharing code and data and software and also archiving it already available today. And there is work ongoing that aims at enabling the legal open sharing of private data. In my field, for example, that's open brain consent, which is a very useful resource. And now finally, I want to highlight the one software solution that I have personally been involved in with software development and that you have seen me use in the very first slides of my talk. This software is called DataLad, and I personally use it to puzzle all of the pieces for computational reproducibility together in my work. DataLad is primarily used in the neurosciences, but it's a completely domain agnostic tool. What it does is data management and data publication, and it is based on two version control tools, Git and Git Annex. And here's what DataLad's main features are. For one, it can version control and link arbitrarily large digital objects. And that is the thing that allows me to version control data or software environments, even if it's hundreds of gigabytes. And then I'm able to link this data in precise versions to the equally version controlled code and results. DataLad has also transport mechanisms for sharing, obtaining, and collaborating on data analysis. And that's why I can publish a data analysis, including all of the components that it involves, to public sources, and then others can go and retrieve it. And importantly, I can do all of that using existing infrastructure. And I don't need any required central service in order to do that. And finally, DataLad allows me to create this machine-readable, re-executable provenance that I have shown you in the beginning of my talk and that has saved my life many, many times. And with those features, DataLad helps me to connect everything that I need to connect. Open source tools, open data, defined software environments, whatever. And the reproducible paper that I showed you is actually a really complex project. It involved, I think, 3 data sets from 2 different sources, not only analysis pipelines, but also source code of other software tools, separate validation analysis, and so forth. But what works for me may very well not work for others. And many other tools and combinations of those tools also exist. And the R Markdown workflow that Andreas will showcase will be a very, very useful thing to have seen at the end of the symposium. But what I really feel strongly about, what is a point that I want to make after having shown you that these tools exist, is that I feel very, very strongly that they are not enough. So we have these tools and services and concepts and methods, but computational reproducibility is inherently difficult and complex and technical. And most of us are not prepped with the skill set to master that during our studies. So it's completely hard. It's super, super difficult. So what we need to do in order to do computationally reproducible science is to empower users to use those tools. And I personally think that those users should turn to these tools not because they must, but because it makes their life easier and their research better. And I think what we need in order to accomplish this are inclusive and collaborative and educational spaces where researchers can, even if they don't have a supportive environment at their institution, find a community that learns together, develops ideas, and improves science together and teaches each other about useful tools. And I personally have found many of those communities. One is the neuroscientific brain hack community. Then there's the community that creates the Turing Way Community Handbook for Reproducible Science. And I'm also building up an own little community in the DataLad Handbook community. And those pictures that I'm showing here, they come from those communities. And I hope that they convey that they are inherently interdisciplinary, completely non-hierarchical, inclusive, and welcoming. And those those things are always a great opportunity for cross-pollination of ideas, for training, for teaching, and for asking questions. And as an early career researcher, these are also the communities where I've gotten the most interesting ideas from. And what I want to stress, a brain hack is not a bunch of bearded nerds in a basement with their Linux computers. It's an interdisciplinary conference with plenty of educational tracks, hackathon-like brainstorming, collaborative sessions, and unconference slots where anyone can talk or teach about a tool or an idea that they have. And if you get the chance to go to a brain hack or another hackathon-like event, then I very much encourage you to do so, even if the word hacking is terrifying. Computer skills of any kind are not a requirement at all to attend. It is completely irrelevant what your skill set or area of interest is. At a brain hack, you will be able to learn, and you will be able to teach, and you will be able to enhance the community with being there. And you can also create a collaborative and interdisciplinary learning environment yourself. What I'm very passionate about also is that I think that everyone should have the opportunity to learn, even if there is no one that can teach them, and even if there's no brain hack to attend. And when I started to join in with software development for DataLed in 2019, I also started an educational community, because standard software documentation is not enough to empower users. I know that because I myself have struggled a lot with learning how to use these tools. So I wanted to create an environment that is not dependent on the presence of knowledgeable people, but that would empower users to confidently use difficult software if they wish to do so. And the resource that I've created is a community handbook. It's an open source educational resource that is aimed at whoever is interested in learning, does not require any pre-existing knowledge about computers or computing, and people all over the world join in to this effort to help others learn. Okay, now that was a really wild ride through all kinds of issues and solutions. And the truth is, computational reproducibility can be really, really hard. But to sum up what I wanted to convey in this talk about computational reproducible neuroscience, I think that we can do it even if we have issues. I think that it is worth it. And I think that everyone should go to a hackathon and learn and help other people learn. And that's already the end of my talk. And I really want to thank advisors and communities and mentors and the awesome psychoinformatics lab that I'm a part of. And I also want to thank you for your attention. You can find my slides if you want to. And I want to quickly pitch the repository for women in neuroscience, because if you want to find or recommend women neuroscientists for conferences, symposia, or collaborations, then you can do this at the repository for women in neuroscience, because women are still an underrepresented group in this field, and you can help to make the field a little bit more diverse. Thank you.
Yeah, thank you so much. And I hope that everybody is clapping at home and sending all these claps to you in some telekinetic manner.
Thanks for the invitation. And I'd like to thank Daniel and Adina for these fascinating talks. I actually have nothing to add. I just reiterate stuff that they said, and it will be, if anything, a little bit more boring, I think. If you wonder about these pineapples, I don't know. I think it's just a pretty picture, but I thought it's like, they are a little bit like reproducible results. Like at first, you know, they look identical, and the longer you look, the more differences there are. And maybe I'm pushing this too much, so let's just start. The work that I'm presenting here is based on the paper that Aaron Peikert and I have written, and you can find this on PsycArchive. And this is really a big shout out to Aaron, who did most of the heavy lifting here. I'm just presenting a lot of ideas that he came up with. And yeah, as Adina and Daniel told us, there are many good reasons for doing science for jointly working towards a joint goal. But really, even if you don't believe in this, it makes a lot of sense just to do it for yourself, just to, you know, set up your analysis in a way that you yourself can reproduce it later on. Because I'm just paraphrasing this from Carl Broman's slides, where he says your closest collaborator is you 6 months ago, but you don't reply to emails. And this makes reproducing analysis really hard. And the sources of failure to reproduce results, and I think I've experienced all of them, I'd like to categorize them into maybe 4 major themes. And one is, in a directory, you'll just find multiple versions of scripts or data. And as Adina has also showed us, that very often there are good reasons that data sets change over time, but often you just have different versions of your script, and it's difficult to find out which one you actually based on your publication on. And then there's also often multiple scripts that all have to be executed in a pipeline, but later on it's unclear in which order they should be executed. You often find copy and paste errors that lead to inconsistencies between the results as they were reported in the manuscript and as they were reproduced or produced on your computer. And then there's this problem of broken software dependencies in the sense that your analysis is broken after an update of your stats package, for example, or there's a missing package dependency that's somehow difficult to obtain or reinstall. And Aaron and I have now implemented or have tried to find solutions for these errors and for these potential problems. And we arranged them into these 4 columns here. And we picked 4 particular tools to address these problems. And for each of these themes here, there are different implementations or different tools. And I'm happy to debate whether there are other tools that are better choices, but these are the ones that worked for us. And the first theme is Git, version control in general, that just tracks the different versions of scripts and data. And the second theme that we implemented with Make is a dependency management approach that somehow documents which things have to be executed in what order. Then there's Docker, that's basically a virtual machine that makes your or encapsulates or containerizes your computational environment such that it's identical even in future or on different computers. And then there's dynamic document generation, which we implemented with R Markdown, that helps us to dynamically generate reports so that we avoid copy and paste mistakes. And I'm going to go into the detail with these things a little bit. And this is just a reminder that reproducibility is even a pre-computer time problem. One famous example is at least 2,000 years old, where this guy told a story and then his followers didn't reproduce it consistently. And this led to a variety of problems later on. And the simple answer, as Adina already has showed us, is any kind of version control system. And we also recommend Git because it's the industry standard, it's tested well, it works really well, it's scalable. And it simply guarantees that code and data are exactly the same version as you used for publication, because what you do with a version control system is that you give a unique identifier to a certain state of your repository. And then you can publish that and reinstantiate this later on. If you use it properly, it also reduces or has the potential to make your code simpler, because it can reduce the number of dead code lines, so code lines that are not used, because it's safe to delete. You always have your previous version, so it's safer to delete than just to uncomment it. And maybe, I don't know your programming style, but what I do very often is that I write a lot of code and then I comment it out and the script gets really long and most of it is actually not working toward the goal or the initial goal. So it's in a way, it encourages deletion of code, and the branching feature, which maintains a separate version in branches, also helps you organize code, for example, code that is stable versus code that's experimental. And then one thing that we like a lot is the releases feature on GitHub, in case you use the GitHub website. Releases, that's something that's used in software. I mean, you know, this sort of software version 0.2 or version 0.1 or whatever, and you can use this for your repository as well. So you can tag it and name it, give it a version and say, this is the submitted version, or this is the resubmitted version, or this is the preprint. And this makes it findable for other people. The one problem with GitHub is that there is strictly no long-term guarantee for the availability of its service. I mean, it's a commercial service provider, even though they do this locks principle, which means lots of copies keep stuff safe. So, for example, they have an abandoned coal mine in the Arctic that they use to store hardware, hard drives with copies of the code. But it's admittedly difficult to retrieve. So what you should do is probably mirror snapshots every now and then, together with metadata and digital object identifiers to other providers that give these guarantees, like Zenodo, Figshare, the Open Science Foundations, and others. This helps making these repositories findable in the fair sense. Then the second thing that I briefly want to touch upon is the dependency management. So the challenge is, once someone found our files in the right version, the question becomes which of those files are executable and in which order. And our solution is the make file, which you're familiar maybe if you're using Linux or Unix. And make files are really simple recipes, basically, or sets of recipes. And each recipe contains its ingredients, that's the dependencies on other files, and then commands to create the product. There's a default recipe, that's the defined entry point for an analysis if nothing else is documented. And by convention, there's an all recipe to create everything that's creatable in a project. And recipes can itself depend on other recipes, so you can cascade down your whole processing pipeline. And why should we use make? Well, because as I said, it has a single well-defined entry point for analysis. So even if you don't ship any documentation, you can download it, run make, and it does sort of the default thing that it's supposed to do. And we can manage all sorts of extra dependencies, even outside our programming language of choice. Things like installing extra software, starting external programs like a pre-processing pipelines, download data from external repositories. And the schema of a makefile is always a recipe name, a colon, then the ingredients, and then the instructions how to build the product. And in practice, this could look like this. There's an all recipe to create manuscript.pdf. So manuscript PDF now itself is a recipe. And it tells us it depends on a certain data set file and a certain manuscript source file. And if these exists, then to build the manuscript PDF, sort of an R script is called to create the manuscript. And if the data file data/iris prep CSV doesn't exist, there's also a recipe for this prepared or processed data file to be created. This is the last two lines in the script. And then there's a different R script to do this. So you have, even if you don't have make installed and you know how to parse or read this make language, it already tells you the dependencies. And then what's even more important together with make probably is Docker or any form of container management. There are others like Singularity that also work really well. These Docker containers can be thought of as basically a shareable virtual machine that runs identically on any computer, whether you run Linux, Mac operating system, or Windows. And there's two ways we can work with these containers. We can either provide a short recipe how to create a container on your machine that's only a few kilobytes, or we can share the entire container. And the container, it's like a sealed, complete package of, you can think of it as the entire operating system. And if you're using R, the R language, or Python, and all the dependent packages in one very large file. This is easily a couple of gigabytes. And these recipes, I mean, we usually try to work with these recipes because it's a little bit more efficient, and there are a couple of nice service providers like the Rocker project that ship these pre-configured Debian images, including R and RStudio. So what this project offers is basically one file that's a whole Debian Linux system, including R and RStudio. And when you download this, you can run your analysis in this virtual machine. And somebody else on a Windows or a different Mac computer or even a cloud computing platform can identically run the same computational environment. And then the other provider that we rely upon is the Microsoft R application network that mirrors the CRAN snapshots and their time machine. And these CRAN snapshots are R packages in a particular version, basically day by day. You can tell the Microsoft network to give you all the packages on a given day in the past. And on the bottom of the page, you see how this recipe for a Docker file would look like. This would tell you something like install a Rocker image with a given R version 3.61 from a certain day, and then run a set of commands. For example, run the install script that installs extra packages, the here package and the Lavan package, and then set the working directory to /home/RStudio within the container. And yes, The easier way that other people have argued for is something like the R package renv that helps you to set up and restore a project-specific local environment in R. And that means you create a private R library with the command renv init, and then you can update this library once you update packages. And renv does basically only save the session information of what packages you loaded and in what version you have them installed. And then with the restore command, you can restore this particular setup that you used at the time point when you ran your analysis and wrote up your manuscript. And that's, of course, a little easier than installing Docker, downloading and building a container, running everything in a container. But we've been arguing that this is sort of probably not enough for some instances. And I just want to give you two examples why this may not be enough. Often we find issues of non-reproducibility that are not within the R environment or the programming environment, but they are system specific or specific to the programming environment itself, but not the dependent packages. For example, if you compare the R version 3.5 and 3.6, and you generate random numbers. What we usually do is we set a seed for the pseudo-random number generator so that the random numbers are random enough, but they reproduce in the same way every time. So here we are sampling the numbers 1 to 10 5 times. What we get in R 3.5 is this set of 5 numbers. If we do it in the updated R version, we get different numbers, even though we use the same seed. And the reason is that there was a bug fix in the random number generator. So strictly speaking, the analysis doesn't reproduce anymore. And this is a somewhat contrived example of computing confidence intervals of a simple regression coefficient estimate with a bootstrap with the identical random seed in both versions. And what you see is that in R 3.5, the 0 is not within the confidence interval, and in R 3.61 it is. And so I don't want to go deeper into issues of frequentist inference and so forth, but I think it's interesting to note that strictly it's not reproduced, even though we can argue it's a replicated result, because they are still compatible with each other. And then there's things like local dependent behavior, for example, English versus Lithuanian computer, that simply have different orders of the Latin letters. So what you see here is the abbreviation of the US states. And if you look at the last row where there's Wyoming and West Virginia, you see that they are actually reversed to the US sorting because there's a different default letter ordering. So this, and if you use this for labeling your graphs, for example, then they may look differently than before. And that's just to make the point that there are a lot of assumptions that we make about our computational environment that may not hold with other computers. And if you use containers, they guarantee identical execution of its content across platforms and time, including, for example, a cloud computing platform. What's also nice with Docker containers is that they can run interactively. So you can run R or RStudio directly in the container, and you don't even notice that. So you see, you get basically your window. You can work in RStudio interactively, but really all your code, even during development time, is executed in the container. And then we can run analysis in different containers. For example, we can run the same analysis with a loop in different containers to see whether any update of a package or R version breaks our code. And this may also help us find out what things influence our results. And the last important piece is the dynamic document generation that helps us or reduces hopefully the number of copy and paste error or mistakes that we make when we transfer results to our final report or manuscript. And for those who haven't seen it, R Markdown is this very simple Markdown language that lets you have both human readable text, your manuscript text fragments, and code fragments in one document. And then there's this very simple, a very simple metadata header on the top left. And then you see these chunks that are this here. I mean, it's kind of a, I don't know, dark green or something. They always start with 3 backticks and end with 3 backticks. And in these chunks, you can have R code. And this R code then gets rendered to a document. On the right-hand side, you see this dynamically generated document that, for example, has a summary of the data or a plot. Oh, sorry, that's just automatically generated. Sorry. Okay. And this is just a great and very consistent way of writing up manuscripts, but also reports. So even if it's just a couple of numbers that you want to send to your collaborator to discuss, I think it's always a very good practice to have this, to use R Markdown for consistency. But even if you're working towards your final manuscripts, there's a couple of nice packages that help you prepare your results in a publication-ready form. For example, there's the Papaya package that helps you with things like number reporting. So on the left-hand side, this would be a Markdown excerpt. For example, the 3 hashtags, that means it's a 3rd level heading. And then you just have plain text on line 115 that says the observed values were. And then you have the single backticks, that's then dynamic code. R tells you this is dynamic R code. You can even have Python code or other different interpreters ready here. So here it says it's R code. And then we call the function print num for print number and the number 1. And we do this 3 times with 3 different numbers. And on the right-hand side, you see this gets rendered to a certain numerical precision that's sort of by the APA style. And you can do the same thing for p-values with the print p command that automatically takes care of the correct rounding and the correct presentation of the values. And it gets even better. For example, if you can't help to run an ANOVA, you can run an ANOVA. Here's just some pseudocode that runs an ANOVA with the affix package. And then what you see is that the result of the ANOVA can be accessed here inside the code. So we have this, again, these dynamic code fragments within the backticks. That's line 146, where it reads results were analyzed using a 2-factor ANOVA. We found no significant effect of gender. Then comes dynamic R code that accesses the ANOVA result. And on the right-hand side, you can see that it automatically renders this into the test statistic, the p-value and effect size estimate. And there's simply no way that you do a copy and paste error here. Unless, of course, you read the wrong data maybe in your code. There's always room for copy and paste errors, but at least given that the code is correct, there's basically no room for copy and paste errors. And the repro package now is an R package that helps you set up all these tools. So all these tools are independent of R mostly, even R Markdown. You can use it with Python only if you want. Even though Python has its own other packages that do similar things. But I think the idea is that even if you have a complex setup of tools and you have pre-processing tools, FSL or whatever, you have to somehow call, maybe some parts are ran in MATLAB. Even this is in principle possible to all have this in a container and have a make file to track the dependencies and then have R Markdown create the manuscript. So I think it's a very flexible workflow. And as long as you're staying in R, or as long as your final manuscript will be an R-based R Markdown file, then the repro package helps you to set up this whole workflow. And an R Markdown file always starts with a relatively simple header that has information like a title, author, date, and the preferred output format. And the repro packages only extends this a little bit with this repro line here that has now 3 different pieces of information under it. That's packages, scripts, and data. And this is something that you manually have to change. So what you can do here is that you just list all the packages that you rely upon in your analysis. You list all the extra R scripts that you want to rely on, and you list the name of all the data files you want to rely on. Then you can load the repro library, and then you can add either all of the tools that I presented or add them one by one. And before you add them, it helps you to check whether your system is set up correctly. For example, you can check whether Git is installed, whether Make is installed, whether Docker is installed, and if not, it gives you pointers of what to do next to install it and set it all up. And once you have done this, there's basically this single command that's called repro automate. And automate now parses the header that you've seen before and then generates the make file and Docker file recipes for you. So what's nice about this, I think, is that it's not very intrusive. So you can even start just coding your analysis without without taking care about reproducibility, and only in the end you automate a given repository. And create these Docker file and make files, and then you can see whether your analysis reproduces in this Docker or make file, and if not, then you can basically explore what are the things that broke. Yeah.
Just as little info, you have about 3 minutes left.
Thank you. And to reproduce a project completely inside a container, a project that you now obtained, for example, from me, you just download this and type rerun. And this rerun executes a make command that builds the Docker file. So it builds from the Docker recipe, it builds the whole image on your machine. And then the second make command here runs the whole analysis inside this Docker container and produces whatever this workflow is supposed to produce. And the results all end up outside the container, basically on your local drive, again, in this directory. Yeah, that's basically all. I think an important thing is that this long-term sustainability is a continuing challenge for the community, and we probably need more academic repositories that are freely accessible, because, for example, GitHub is strictly a commercial provider, or is at least run by commercial companies, the background. And these academic repositories should guarantee long-term reproducibility independent of short-term grants that we often have. And they should not only provide disk space. I mean, we are at a stage where a lot of them now providing disk space for us, but they should also provide software infrastructure and computational environments so that we can run these things online. And some providers are starting this, but I think we need more. And generally, I think we should just better recognize the efforts of maintaining scientific software and repositories, and giving more incentives to people to share their data, and also give them credits for creating digital collections of whatever this is, collections of software, collections of data, collections of scripts, and so forth. And with this, I'd like to thank you. If you want to know more, you can ask a specific question about the workflow via a GitHub issue. That's our discussion forum, and where we try to interact with all the users. You can follow me on Twitter. You can read our preprint. You can contribute to our repro package via GitHub. And there's also a different talk by Aaron on the same topic. Yeah, thank you. I'm looking forward to your questions.
Great, thank you for the talk. I can also double down and say reading the preprint for me personally helped a lot. Yeah, so I thought it was a very good starting point.